record the lecture to see if we can, just in case someone is quarantined or missed, I'll start sending these out. And then hopefully that'll help. Now, I believe we did a little bit of the DNA replication, but I know that's right around where we stopped. And this is where I'm just going to start with this again. All right, so the end of chapter 4 ends up being about DNA. I wish that wouldn't slide. Anyway, and how it is used to make proteins and different things. And so the first part we look at is just talking about the general idea of it. It says it's needed to synthesize proteins, which are a lot of them are going to be the enzymes that we just talked about in a previous and earlier in this one. Now, in DNA, the entire genetic code is called a genome. Right? All of the, if it was a recipe book, the book would be the genome. Each individual recipe is considered a gene. Right? And so, in general, these are going to be used to make proteins. Now, the portion of the genome that encodes for proteins is called the exome, and it's still a small part. The DNA is a really amazing molecule, and we're still trying to figure it out. You know, people talk about how much we know about it, but understand that when the egg and the sperm get together, there's no other information that enters. That DNA is what controls everything. So if you're looking, when you're studying those bones, every bone has little holes, ridges, and everything. That's controlled by DNA. Yes? Oh, it shouldn't be. I just tested it, but. Is it this red? It might be the big. We'll see. No, we'll that. And if it is, I'll figure it out. But I just did a short one to check it, and it said it was on. I mean, it worked. So, but then again, it's the way things are. So you never know. So, maybe I'll put maybe I'll get some subtitles and whatever. If it doesn't work, figure it out. But anyway, so again, the DNA is far more complicated than just protein synthesis, but that's the main thing, and that's what we look at. Now, DNA, as most people know, is this double helix form. If you've looked at it at all, it's this little twisting spiral ladder type thing, and it is going to be connected by what we call base pairs. And these bases, adenine, guanine, cytosine, and thymine, are the bases for DNA. And it is always going to be where adenine binds with thymine and guanine binds with cytosine. And it doesn't matter which side it's on. If I've got an A on this leg, there's a T on the other side. If I have a T on this leg, there's an A on the other side. All right? On DNA, that's the way it works. And they're called complementary base pairs. And so when we look at it, this is the twisting ladder, and you can see, I mean, it doesn't matter which side it's on. A and T always go together, and C and G always go together. And if you remember, these little dots represent hydrogen bonds. Right? The key to DNA is its ability to unwind, be read, and then be put back together without much trouble. And it's all because of these hydrogen bonds, these little refrigerator magnets that we kind of talked about. Now, when cells go through mitosis, at the end of mitosis, when we looked at it, we talked about how the end of mitosis, I've got two daughter cells that have identical DNA, identical genetic material. Now, the two daughter cells might have different cytoplasm and organelles, but they have identical DNA. And it's because during this interphase, we make a copy of it. And it is called DNA replication. So right now, especially in your skin, that little layer under the epidermis is constantly going through mitosis. Right now, again, DNA is about a 6-foot-long molecule anyway in real life. All through those little cells underneath your skin, they're making a copy of it, preparing it, making cells, and putting up. So, I mean, it's pretty amazing that it can do that quickly. But the DNA double helix will separate the hydrogen bonds between the base pairs, or what allows it to do that. It unwinds. And this real special enzyme called DNA polymerase goes in and it organizes the second leg. So when we're going through and we're looking at it, it's going to split. The original DNA is going to open up, and then we're going to have that DNA polymerase come through and replace the other side. So we end up with two strands of DNA now, and each strand is going to have one original leg and one recreated leg. All right, so the two daughter cells will have that. They'll go through. Once they get into interphase, each one will make a copy of it. Each one will have an original strand and then a copied strand. And so we call this DNA replication. All right. Does that make sense? Now, this part is where I believe we left off last week. So protein synthesis. Now, as soon as you hear this, I hope, and not everybody's going to think this way, but I hope that at least there's a few of you that are like protein synthesis ribosome. Because ribosome we talked about is about protein synthesis, right? And so protein synthesis, making proteins, the recipe for the protein is in the DNA. Right? We call that a gene. Now, when we look at this, it says it's the genetic code. When we look at this, it is about these base pairs. Every three base pairs, every three nucleotides on the DNA represents an amino acid. So, and we'll see this in just a second. So if we're looking at the DNA and we're saying, all right, this is, on one strand, this is A, this is T, this is A, G, C, whatever. Each group of three represents what would be one amino acid in a protein. And remember, proteins are long strands of amino acids. So it's the recipe for it. Now, there are nucleotides that we're going to see that are going to be groups of three that will represent stop and start. I hope you would know that if I say, what does the stop code cause things to do, you would figure out that it's stop. Right? Start is start. Right? All right. Now, in the process of this, there's a two-step system. One is called transcription and the other is called translation. Transcription literally means to write down. If you're writing notes, you are transcribing. The word transcription means to make a copy. You write it down. Old time, before computers and stuff, secretaries were called transcriptionists. Their job was, if the boss wanted to dictate a letter, they would go and they'd write it down. And that's where the word comes from, transcription. And that's what we're going to see. A copy is made. It's written down. And then we have to translate it. Translation means taking something that's in an inusable form and turning it into a usable form. If I gave y'all the test now and had it in Mandarin Chinese, which I'm not assuming that many people speak, you would take it and have to translate it to make it useful. All right? And so we're going to see how this happens. Translation, we're going to see happens on the ribosomes. This is when the little codes that are copied turn into amino acids and into a protein. That's translation, happens on the ribosomes. Transcription happens in the nucleus. It is what we're going to look at. The nucleus, again, is this little walled system. We talked about how the DNA, the chromatin that's in there, cannot come in contact with the stuff in the cytoplasm or it will destroy it. And so we've got a recipe in this little vault, and so we have to copy it down to leave because the recipe itself can't leave. So we start off with transcription. All right, as it says, DNA molecules cannot leave the nucleus. So since the protein synthesis, the making of the protein happens in the cytoplasm, we have to have some carrier molecule. And what we're going to see is the carrier molecule is called messenger RNA. Messenger RNA is carrying the message. Right? So messenger RNA, or just RNA in general, are single-stranded, which is different than DNA, which is double-stranded. Obviously, the RNA is going to be much shorter than DNA because DNA has got all of the recipes on it. So if I had I don't know how many people like to cook, but a good cookbook is called The Joy of Cooking. The Joy of Cooking's got like 800 recipes in it. I don't have to copy down every one if I want to know how to make a buttermilk biscuit. I just copy down buttermilk biscuit, right? And so RNA, which is making a copy of the individual recipe, is going to be shorter than DNA. If I make a copy of how to make, buttermilk biscuits, I don't have to write the whole book down. So the messenger RNA is what is going to go through the process of transcription. Now, just like we had DNA polymerase, which goes through and makes the second leg of the DNA when we make the copy of DNA and replication, we're going to have RNA polymerase, which is going to create this messenger RNA. Now, again, as we get to the next couple of slides, I'm hoping that I'm going to start writing some stuff down to show you what's going on. Now again, RNA polymerase, as it says, recognizes the begin and end of a particular gene. So I've got this big strand of DNA. I need to make protein X. It's an enzyme. My body knows what section of that long strand is representing protein X. It will take it and it will unwind it. It'll take that little section, it's all twisted, it'll untwist it and open it up. RNA polymerase comes through and starts assembling RNA in the middle of it. And what it's going to do, now I've got two legs. What it's going to do is it's going to read one leg. It can get very complicated, which one it reads and whatever. Right now we're just worried about it's going to read one side. And it's going to start making a copy. But there's going to be a big difference. All right? In this, messenger RNA does not have one of the base pairs. Thymine, which is found on DNA, is not on RNA. It's everything else is the same except this. Wherever there would be a thymine, it is uracil that's on the other side. All right? Now, that only affects if it's thymine on that side. So if I'm reading this copy and this is the DNA and I'm making a copy, if there's an A, there should be a T here and there's a U. But if there's a T over on the other side, there's still an A. That's normal. The only change is anywhere there would be a T, thymine, on the messenger RNA, it's a U for uracil. Does that make sense? All right, we'll go through this. I don't want to skip by that. But there's some differences on here. These aren't like huge. I'm hoping you know that DNA is deoxyribonucleic acid, and so deoxyribose is the sugar for DNA. Ribonucleic acid is RNA, so it's ribose is the sugar. Again, not something I'm super, gonna beat into your head. There might be one question maybe on that. It's not a huge deal for me. I'm hoping that you know DNA and RNA. Right? DNA, I want you to know, is double-stranded. RNA is single-stranded. That's, to me, is far more important to understand in this. Now, on this, and I'm going to pull this up for a second. So on this, if this was one strand of DNA, I'm saying that these are them over here, the other strand would be this. Again, this is pretty simplistic. Wherever there's an A, hydrogen bonds, I'll draw it with hydrogen bonds. Now, when this unwinds and I have this messenger RNA get in the middle of it, it's going to make a copy. The big change is the copy over here is going to be a U because I don't have T on the RNA. Everything else ends up being the same, right? So if I had something that looked like this, right, and I said, all right, I'm going to put three little sections. That would be 3 amino acids. If this is DNA, so I'm going to put the other strand of DNA down here. Right? What would this first one be on this side? Did everybody see that? I mean, we went pretty quick, but there's going to be a question on this test that I'm going to have one string of letters. I'm going to say in DNA, tell me the other strand. You'll have 4 choices, right? But you're good with that? If I was saying, all right, now I'm looking at the messenger RNA. So I'm going to put this little line in there representing the messenger RNA, and this is what it's reading, right? So really, it's the same as this, except wherever there's T's, it's just a U. So everything else is the same. So it's not, don't overthink it. Do one, like if I'm looking at DNA, just do one. And if you're looking for messenger RNA, sometimes it's easiest just to go ahead and do the DNA and then say, all right, anywhere there's a T, it's a U. There'll be a question about that with the RNA. I'll give you a string of letters and say, all right, I need you to tell me what the base pairs would be on the RNA. Do you feel all right with that? And if you don't, look, I understand if everybody else is getting it and you're not, you don't want to go, hey, I don't get it. Send me an email or something. We'll work through it. All right. All right. Now, in the DNA and the messenger RNA, these are called codons. Each group of 3 is called a codon. All right, so even in the messenger RNA, that's 2. Now, like a molecule like hemoglobin, I want to say is like, gosh, now I'm going to butcher it. I want to say it's like 400 amino acids long. It might be something around 300, whatever. But let's say it's 300 amino acids long. That means that it is 900 individual little letters long. Yes?
So specifically, codon is the amino acids in that line?
It is. It's not it's what these three represent what's called a codon. Okay. They're called a codon. They represent what will eventually be amino acid.
Okay, so
Does it?
Is an amino acid like the chain of those?
We'll see. I'll show you in just a second. All right. But I know what we'll get to that in just a second, because what's happening is now I've made this copy, messenger RNA. All right. DNA, these 2 strands cannot leave the nucleus, and the ribosomes are out in the cytoplasm. So this messenger RNA is now going to leave. It's going to go out through a nuclear pore and find a ribosome. This will connect back together, twist up, and go back to normal. Right? The book closes. Now I have this string of codons in the messenger RNA. It is now going to find a free ribosome. Ribosomes, well, I'll erase this and come back to it. Ribosomes are a very special little organelle that actually are made up of another form of RNA called ribosomal RNA, rRNA. Now again, ribosomal RNA is about ribosomes. It takes 2 of them. Now, as this leaves and it finds this ribosome, it will start the process of translation. So if I go again and say, all right, this is my little messenger RNA, and I say, and I'm just going to throw things out here. Oh, it can't be T. Hey, let me U again, whatever. UCG, whatever. So this would represent 5 codons. Each one of these represents some sort of amino acid. I have to translate it from this language to amino acids to make a protein. Does that make sense? This is like Braille, and I can't read Braille, so I need somebody to translate it into written words so I can read it. And they're 2 different things. This is completely different than what the amino acids are. Amino acids are a pretty complicated molecule. Right? So what happens is, as this goes into the ribosome, and we'll see this in just a second, the ribosome is set up where it's almost going to have this little skylight part to it. And it's going to start taking this through here, and it will have it, and it'll stop briefly where these codons, each codon will stop underneath this skylight. Now another type of RNA steps in at this point, and it is called transfer RNA, tRNA. Its job is, and it's even smaller than the messenger RNA, it looks into this skylight and makes a copy of what that codon is. So it looks at it and it says, all right, I got these 3 base pairs. It makes a copy, and we call that copy an anticodon, right? Because it's, again, I've changed it a little bit. It's going to be, you know, where if this is an A or a U or a C, it's going to be the opposite on the other side. So we call it an anticodon. Now there's 20 different types of amino acids that make up our proteins. And so imagine I've got a box over here of Legos, and I've got 20 different colors of Legos. All right? Messenger RNA starts being pulled through the ribosome. The transfer RNA is going to read and make a copy of the anticodon, and each one of those colors is going to have an anticodon that represents it. So the transfer RNA is going to read it, it's going to go over to my box of Legos and say, all right, it's yellow. I'm going to get the yellow Lego. It's going to come back over, it transfers it into place. It puts it in place. Now the next group gets pulled through, and now I have the next codon in here. Transfer RNA reads makes an anticodon, goes back to the Lego box and says, all right, this time it's blue. I'm going to take the blue Lego and put it into place. It transfers it back into place. And as it's doing it, it continues to snap these together and make a long chain of amino acids that we end up calling a protein. It is translating it from this unusable form to one of those weird shaped enzymes we talked about. Does that make sense? Somewhat? Did that kind of make, because it relates to what you were asking, I think, right? And so, I mean, it's crazy that it does this. Yes?
Is it like positive and negative? The positive codon will attract to the negative codon?
What will end up happening, and this, we're not going to get too much into this. All right, give me 1 sec. It won't be on the test. No, I'll show you what's going to happen here, because it's pretty interesting. So again, as this says, and I'll pull this down again, I see that the recording's going to be all whited out when it's not. Come on, stay there. Stay there. I was going to get frustrated on that one. So, inside the nucleus, DNA, whole bunch of different genes in this long strand. A particular gene I need to make a copy of, my body makes messenger RNA, it gets in the middle of it, and it makes a copy. The biggest difference is, in that copy, wherever there would be a thymine, I use a uracil instead. Messenger RNA then leaves. Now, making the copy is called transcription, which literally means making a copy. As it leaves, it'll go through a nuclear pore, find a ribosome. In the ribosome, those little 3 groups of bases, called codons, get read by something called a transfer RNA. It makes an anticodon, and then goes and gets the amino acid that anticodon represents and puts it into place. So it's going from these little base pairs that don't mean anything to making this string of amino acids that end up having weird shapes. So we've gone from this thing we can't use to something we can. That is called translation. Translation happens on the ribosome. Right. Now, I've got a couple other ways to describe it. We'll see. Now, again, as this is saying, the messenger RNA, the ribosome holds the messenger RNA in place while the transfer RNA carries the correct amino acid into the sequence. As it goes through, again, since we are making a big molecule, we're building. It is dehydration synthesis that we use. Remember, we talked about anabolism is made by dehydration synthesis. I join things or synthesize things by removing water. If I was breaking it down, it would be hydrolysis, hydrolysis, right? Now, as it starts being put together, what ends up happening, you're talking about the shape, about positive and negative, it's more along the lines of some amino acids are water soluble, which means they're charged, and some are lipid soluble, which means they're not charged. And when you put them together, they start folding or twisting. They become attracted to each other or repel each other. And that ends up causing it to have a weird shape, spirals and folds and different things, and they're very specific. And we call that conformation. That is, each protein has a unique shape, and we call that conformation. That's what we talked about. If pH denatures a protein, it's lost its conformation. It's lost its unique shape. It can no longer function. Now, this is, if you ever need, help going to sleep, you might want to look through this. I will not test you on any of this. This is kind of going through what each of the kind of codons mean or what they represent. It goes over, this is the 20 different amino acids. Again, not something I'm worried about. But this picture shows both steps. This is inside the nucleus, and this is what we call transcription. This is unwound, it opens up, messenger RNAs come in, and it's made little copies of this. Rna Polymerase is going through and writing it down. Again, this and this would be exactly the same except for the T and the U. Right? There's a U on messenger RNA. When it's done, it's going to leave through one of these little nuclear pores and find a ribosome. Now, when I was a kid, Evel Knievel was a big deal back in the 70s. And I had one of the coolest toys. It was one of those little Evel Knievel motorcycles. You put it on a little bait and wind it up. On my brothers, though, they would do that and then they'd stick it on my arm, It's like, ah. But it also had what I called a rip cord. It was a plastic little cord, and on one side it had teeth. The other side it was flat. You'd put it through the wheel and you pull it, and then you set it on the ground and it'd take off. Right? Everybody kind of can imagine that. You know what I'm talking about? That's what this basically is. Messenger RNA is like a ripcord. It's got these little base pairs in it, and they're groups of 3. And so kind of like I would feed that ripcord through the wheel, this gets fed through like little teeth through the ribosome. Each little group of 3 stops for just a second, long enough to let this little thing called transfer RNA read it, make a copy, go get the amino acid, and come back. And as it's keeping to do this, it keeps adding one brick after another, and it ends up making a long chain of amino acids that once it gets done, now it is useful. So I've translated it from something I couldn't use. This does, if this was hemoglobin, this doesn't hold oxygen, right? I couldn't use it. So I've got to translate it into a usable thing. So the way that this ends up being a shape that allows for oxygen to be trapped on it and held in it until it gets to the tissues. So I go from one that I can't use to one that I can. Right? Translate it. Yeah.
Like the peptide chain?
Yes.
Picture right there?
Yes.
What happens to everything once it goes through?
What happens to this?
Yeah.
It gets broken down, recycled, and brought in here so that we can use it again. Yep.
That figure is 414.
Okay.
You didn't add that on there.
I did. I'm sorry, I didn't have this one on?
Yeah, that's 414.
Okay.
Figure 414. Your other pictures have which tables they were.
Okay. Oh. I didn't mean to. No, I didn't. Now this is kind of showing, it again. It's saying, all right, it's pulling it through. It's kind of giving a little bit of a more in-depth picture of the translation. But again, the big thing is messenger RNA is the one that does transcription. Transcription happens in the nucleus. Translation happens in the cytoplasm on a ribosome. It requires transfer RNA to put the pieces in place. So it's kind of like, I know this is weird, but Kentucky Fried Chicken. Yeah. Kentucky Fried Chicken. They actually have as far as I know, it hasn't changed in the last couple of years, there is a vault in the main headquarters in Kentucky that hold the actual recipe of the secret 11 herbs and spices, which I think everybody knows what they are, kind of, whatever. But they don't allow, they only allow, when you buy a franchise, they only allow like one person to go in and get trained on how to do things. So the nucleus is kind of like that vault with the little recipe in it. It'll allow one person in and they can copy down the recipe. Now, if you're familiar with the way restaurants work, if you're in a restaurant that has a quote unquote chef, you have what's called sous chefs, people that do all the grunt work basically. They're the ones that dice up things and whatever. That's like transfer RNA. The chef is kind of like the ribosome in a way, saying, all right, get me onions. I need some shrimp. And the transfer RNA are the ones that go get it and bring it back. Does that make sense? That's how it works. And again, this is, it's going on all the time for every type of protein, every enzyme that's created in your body. This is what your body's doing. I mean, just in the time we've been in here, this has been going on in, hundreds of thousands of times in different cells of yours right now. Thank God we don't have to know how the body works for it to work. That would stink. Yeah, there wouldn't be many alive, that's for sure. I know I wouldn't be. So, at the very end, now we're gonna talk a little bit about this. This is not something I'm going to test you on. But I hope you understand it because this ties in with the primary, secondary, tertiary, quaternary view of proteins. So a protein has to have a certain folded look to it. Different ways that it is bent and twisted, and each one is unique. We call it conformation, right? So there's a part of our body that its whole job is to go through like somebody making a little balloon animal, ring, 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 and twisting this and making sure it's in the right position so that it works. And we call those protozomes. Now, if it doesn't fold correctly, these also tear it down. So basically, they're folding it, and it's like, again, a balloon animal. It doesn't work. That doesn't look like a dog. Pop, get rid of it. Let's start over. Right? Now, once it has been turned into a protein, this whole process is said or described as the protein is being expressed. It has gone from something that's just an idea, a thought, a gene, to actually being something real, hemoglobin, whatever. It has been expressed. Right? Now, in each cell, like again, every cell in your body that has DNA, it's the same DNA. So the DNA that's controlling your eye, for instance, is the same DNA that's controlling your fingernails. And it's the same DNA that's controlling your synovial fluid in your joints. Different areas will have different expressions of their genes. Right? The little cells in the kidneys that are going through and learning, filtering out things, knowing what to bring in, are the same things that are in your lungs. It's just the different expressions cause different genes to be made. Yeah. What happens, now I could be wrong, and I'll tell you, I am not educated on that. So, but I think what I understand is, it's like, here, the very first slide on, I think it's this, has something called arsenic, which is one of the most deadly poisons that we have. And in a way, it kind of is going to go what I think happens with mad cow disease. Arsenic is a type of amino acid. There's only a few amino acids in the world that aren't found in our body. And they end up being poisons, because what they do is when the body starts putting these together, they can end up sharing a similar, I could use this, a similar codon as what we really need. And then our body will grab the wrong thing and put it in, and it will cause a malformation of the protein. It won't, even if it's just one brick, it can change the entire structure of the protein. And I think that's what mad cow disease does, is it alters certain amino acids to where they cannot do their normal thing. And it changes the enzyme shapes, and the enzymes don't work. But I could be wrong on that. But that's what like arsenic does. Because again, it's pretty strange how fine-tuned it is. Just a little bit off one way or the other, and you know, it's death. So that's why it's really kind of important, because death is never a good thing. You know, I mean, yes. Yeah. Interesting. There you go. I knew it had something to do with protein, you know, how it affected the proteins. I thought it was affecting the amino acids, but it's saying it's a protein on its own, and it just goes like Pac-Man in our body and it's not happy. Yeah. Interesting. Yeah. I mean, and again, it's funny how many things, really, how many things there are out there that want to do us harm. You know, and how easy it is, but how, you know, I mean, again, I don't mean this mean, I'm not saying, you know, not to, you know, I'm not saying anything about COVID or whatever, but man, our body's pretty, don't ever lose sight of how amazing our body is. If we put it in the right spot, it can fight most anything. But again, you know, you got to be smart about stuff. All right, so that is the end of chapter 4. So this test is going to be 3 and 4. Now I was going to say, you know, we'll do the test on Thursday. I think that's what we talked about last week. I'll be honest with you, I haven't had a whole lot of time to take advantage of making the test like I want to. It's been a while since I've taught 112, and my old tests, they're not me anymore, you know. And so, again, we are kind of, it's my fault as the conductor of this train. I've kind of put us a little bit behind. And so we're going to pick up the pace a little bit. But this next test, again, we're going to do a review on Thursday. It'll be Tuesday that we have it. And then we'll start, you know, hightailing it through the rest of the way. So, any questions about anything? Did everybody get the email I sent out with the videos? I sent a playlist with the videos. You know, I'm going to try, again, audio. I did that with chapter 3, and I've never had a problem with it before. I had a problem with the chapter 3 audio on the PowerPoint, so I'm going to redo those and send them out. But, you know, so Tuesday or Thursday, gosh, we will do a review, get it kind of set up. Do not overlook the connect. All right, I'm going to go check and make sure that it is up. I believe it is. But please do the connect. It will help you study, right? It'll be in your best interest anyway, but it helps you study. And so please try to do that before Thursday, or at least play around with it. So if you haven't done it, if you have questions on Thursday, we can answer it. After Thursday, you know, I do not work on computer problems over email. That never works out well. Yes?